Hi, everyone. Welcome to OsteoBites, and thanks so much for joining us today. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Amanda Marinoff from UCSF, and she will be discussing clinical biomarkers for osteosarcoma stratification, or cBOSS for short, insights from a working group. Thank you so much, Dr. Marinoff, for joining us on OsteoBites today. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom. It's one of the buttons at the bottom, and we will share those with Dr. Marinoff after her presentation. A little bit more about our guest today before we jump in. Dr. Amanda Marinoff is a pediatric oncologist and translational researcher at UCSF, where she focuses on developing molecular biomarkers to improve risk stratification and treatment for children and young adults with osteosarcoma. She co-leads the International Clinical Biomarkers for Osteosarcoma Stratification, or cBOSS for short, initiative which brings together experts across North America and Europe to evaluate and prioritize emerging classifiers for clinical use. Her research aims to bridge the gap between genomic discovery and therapeutic application, advancing precision medicine approaches for patients with high-risk disease. Dr. Marinoff earned her medical degree from Harvard Medical School, completed her pediatrics residency at Boston Children's Hospital, and completed her pediatric hematology oncology fellowship at UCSF Benioff Children's Hospital. She's an active member of the Pediatric Solid Tumor and Early Phase Clinical Trials Group at UCSF. We're thrilled to have you in OsteoBites today, Dr. Marinoff. Thank you so much. We just have one announcement before we get started. It's August tomorrow already, and so that means we're really preparing for September Childhood Cancer Awareness Month. And as we do every year, we do gold bows, so you can help out by signing up as a bow agent volunteer. And as a bow agent, you'll help raise awareness by distributing beautiful gold bows throughout your community on mailboxes, storefronts, schools, and more. Each gold bow is a powerful symbol of hope, resilience, and solidarity for children and families facing cancer. So help us turn neighborhoods gold and make a meaningful impact one bow at a time. And I'll put a link to the forum to get more information in the chat. And that's kind of it for me. We'll go ahead and get started with our presentation today. Dr. Marinoff, if you want to go and start sharing your slides.
Absolutely. Thank you so much for the kind introduction, Christina, and for the invitation today to share our work on cBOSS, which you beautifully said. It's a mouthful. It stands for Clinical Biomarkers for Osteosarcoma Stratification. Are you all able to see the slides in the right view?
Yes, that looks great.
Wonderful. Many people for years have really recognized the need to advance precision oncology in osteosarcoma. And it really was at Factor 2024 when I had the opportunity to sit down in Cleveland with Ryan Roberts and Andy Livingston, and we said, let's make it happen. Let's create this working group and work on prioritizing biomarkers and classifiers with a focus on clinical translation. And that was really how cBOSS was born. I'll start by sharing my disclosures. First, I want to make it better for children with osteosarcoma and their families. And second, I believe we can and will. And with that, I just want to start by anchoring us in the clinical problem that really motivated this work. We know that osteosarcoma is extremely clinically and biologically heterogeneous, and yet our treatment remains one size fits all. This is a plot where we can see each of these are called circos plots, which represents the very complex and chaotic genome of osteosarcoma, each of these representing an individual osteosarcoma. And what you can appreciate is each of them looks different. No two are alike. And yet every patient, essentially at the time of diagnosis, receives the same therapy, MAP followed by surgery, followed by more MAP therapy, regardless of their initial clinical presentation or underlying biology. Now, this is not because we don't have candidate prognostic biomarkers that are able to stratify osteosarcoma by survival. This very busy slide is actually a Google search for osteosarcoma stratification. And what you can see is so many Kaplan-Meier curves or survival curves that you can really wallpaper the office, as Ryan Roberts likes to say, with the number of survival curves that we have. And yet, despite really the explosion of publications and nominated classifiers to stratify osteosarcoma, none of these are currently used in the clinical setting. And of course, the questions are why, and that was really the motivation for forming cBOSS. Our goals for our working group were to evaluate the current landscape of prognostic and a subset of predictive biomarkers that were associated with clinical outcome, to build an initial framework for clinical risk stratification. And this effort by no means looked at every single prognostic or predictive biomarker that has potential, but we really wanted to hone in on the ones that had potential for near-term clinical use. We wanted to identify the evidence gaps and unmet needs and create a roadmap that would allow us to align future trials and correlative studies around shared priorities. And our approach was leveraging Zoom. So we used 9 virtual sessions from August to November 2024, with experts joining from across North America and Europe. And for each nominated classifier, participants with the deepest domain expertise reviewed the literature, really with an eye toward the biological rationale for each molecular feature or classifier, the strength of the evidence across data sets, the clinical and technical feasibility of implementing these, and the readiness for and gaps to translation. Here is our team. It was really an absolute privilege to have the opportunity to work with this phenomenal team, again, spanning many countries and I think 10 or 11 time zones. And so that meant very, very early or very, very late Zoom calls for participants that were either on either end of those time zones. And I also want to just mention that there are many, many, many other wonderful clinicians and researchers who are doing really important work on advancing prognostic features and markers for precision oncology and osteosarcoma. And so this is really just a subset of those who are doing this work. Here's an overview of the features that we evaluated that were broken up into a few different subtypes. First, we looked at clinical features, and then we looked at molecular features focused on prognosis or association with clinical outcome across 6 domains: circulating analytes, primarily circulating tumor DNA, DNA-based changes, looking at copy number alterations and loss of heterozygosity, transcriptomic features, epigenetic features, and tumor microenvironmental features. And then we looked at a subset of molecular features that could potentially be used as targets for targeted therapies, with a focus on the surfaceome and the DNA damage repair pathway. We sought to try and take this really vast set of classifiers and a really vast and growing literature and try and put it into something that we could hold and use as a basis for our starting point in terms of where we stand for each of these biomarkers today and what the gaps are to move these into the clinic for implementation. And so we gave each of these molecular features that we evaluated a classification ranging from 1 to 4. 1 is needs further preclinical evaluation. So these are classifiers or molecular features that have promising data, but the evidence right now is primarily limited to preclinical models or exploratory human cohorts. The second category is needs replication. And so these were features where we saw an association with clinical outcome in a single or small patient cohorts, and we need to reproduce these findings in additional or larger cohorts. The third is needs prospective testing with a predefined statistical plan. And so these are features where we've seen an association with clinical outcome across multiple retrospective data sets. But in order to move this into the clinic, we need prospective testing, again, with a predefined statistical plan and threshold for defining the biomarker. And the fourth category is ready for clinical use or to define eligibility for a subgroup on a clinical trial today. And those are features that have already passed this test of prospective testing with a predefined statistical plan. Before I dive into the molecular features, I want to take a couple of steps back and remind ourselves that clinical features provide really important prognostic information. Before we even dive into the really amazing science that's emerging, we have a lot to work with already. We already know, and these are just 3 representative studies of many, many studies that show that stage, primary tumor site, and resectability are all very highly associated with prognosis, with the presence of metastatic disease being, at the time of diagnosis, being our strongest prognostic factor, patients with pelvic primary tumors having a worse prognosis, and patients who do not have resectable primary tumors also having significantly worse prognosis. And the question here is, we have this strong foundation, and can we combine these clinical features with molecular features to bring precision medicine to osteosarcoma? It's possible someday molecular features will surpass these clinical features, but right now, these are really critical tools that we have available to us, and we think that the combination with the molecular classifiers will allow us to really deliver the most robust precision medicine. And then, of course, histologic response to chemotherapy, which is one of the most well-known prognostic factors that we do utilize to some extent in our clinical care. It's something that we report, that patients and families ask about, and that we talk about in our tumor board. But the question is, how useful is histologic response to chemotherapy, or the percent necrosis? And certainly there are many retrospective and prospective studies that show that there is an association with survival. And yet, we really know that this is a suboptimal marker of response. We know that there are some patients who have greater than 90% necrosis and end up having relapse disease and poor outcomes despite a biomarker that told us that they should do well. And on the other hand, there are some patients who have very poor percent necrosis when at the time of resection who end up surviving and doing well. These are a couple of studies that show us the other limitations associated with histologic response. One, we know that dose intensification of chemotherapy actually leads to increased necrosis, but not increased survival. And so conflating the two is imperfect. And secondly, we have not been able to improve outcomes by changing therapy based on poor percent necrosis, and that was demonstrated in the URAMOS trial. And so we know that it's time to do better, and that we need biomarkers or molecular features that tell us how a patient is likely to respond to therapy at the time of diagnosis, and not when we're more than a third of the way in, and that's really grounded in biology. Here is our cBOSS wallpaper, and I'm going to go through each of these panels and then do a deep dive into a couple of the molecular classifiers that we took a look at. Again, we know that there are many candidate prognostic markers, and I'll walk us through briefly A through H. A, again, is the presence of metastasis at diagnosis. B is looking at circulating tumor DNA, which is one that we'll do a deep dive on in a few slides from now. MYC amplification is another molecular feature that we'll do a deeper dive on. Genome-wide loss of heterozygosity is demonstrated in D. E through G are 3 different transcriptomic classifiers, and H is looking at overall survival by methylation profiling. And when we zoom out, we can see 8 different classifiers of the many that are out there, where we can see separation of the survival curves based on the group. And the separation of the curves tells us it's pretty good at distinguishing who are the patients who are likely to do well from those who are not likely to do well. And yet, again, we're not yet clinically using these because there's a lot of questions that remain. How do all of these 8 classifiers and the others that are out there, how do they fit together? Do they provide overlapping information? Are they complementary to one another? Which tests do we use to figure these out? And how would we actually implement it such that we can improve survival and reduce treatment-related toxicity for our patients? This is a question that, I think many of us are spending our lives working on, and cBOSS has not addressed in its entirety, but I think we've provided a foundation and a good start. Here, I think we've started by trying to address what is the state of each biomarker currently. And this is a very high-level summary of each of these, and I'll go through briefly, and very happy to take questions on any of these or other emerging biomarkers that folks want to talk about in the Q&A, but I won't do a full literature review of each of these. But in summary, we found, we showed again that clinical features, including stage, resectability, and primary tumor sites, going back to basics, these are ready for clinical use. That seems obvious, but we're not yet using it to differentiate therapy. Pre-treatment circulating tumor DNA burden is also ready for clinical use. MYC amplification, CDK4 amplification, and genome-wide loss of heterozygosity are all in need of prospective testing with a predefined statistical plan. There are many emerging transcriptomic classifiers in the literature that show promise and an ability to stratify survival in patients. And we're highlighting 3 here that we think are sort of closest to translation because they've been reproduced in several data sets and use methods that improve their accuracy and reduce bias in these. And this is one classifier called OTTR, which comes from the Sick Kids group in Toronto, a set of signatures named G1 and G2 by the French group led by Antonin Marché. And just a few days ago, Emanuela Palmerini and group, also looking at data from the French OS 2006 trial, published a 21 gene prognostic signature that was validated in multiple retrospective cohorts. There are several other molecular features that we think show promise in small or single data sets that need to demonstrate reproducibility in additional cohorts, and that includes methylation. Work that's being done by Dimitris Bentos at MGH is highly promising, and they're working to validate those right now. There are a number of additional copy number alterations. Here are just a few that I put on the screen. Immune composition, there are several markers there that again, show early promise but need replication. And then also emerging preclinical data showing that chromatin conformation that can be detected by attack sequencing can differentiate different biological subgroups of osteosarcoma, and structural variant patterns are emerging as well. And these are in early days, but show promise for being able to be used in the future. That was a bit of a whirlwind, and I want to sort of now go into a couple of these biomarkers to show you our process and how we're thinking about this, and why some of these are green and why some of these are blue, and what the gaps remaining are that we need to address to really be able to use them. Again, this ready for clinical use is based on having been demonstrated in a prospective cohort with a predefined statistical plan. And so we just had one molecular feature that was pre-treatment circulating tumor DNA burden that met that bar today. And I'll show you a snapshot of that data here. This is work from the prospective leopard study led by Dr. Dave Shulman at Dana-Farber. He's presented in a number of settings before, including OsteoBites, I believe. And his study has demonstrated that elevated baseline circulating tumor DNA burden is associated with inferior event-free survival in patients with localized osteosarcoma. And that was demonstrated with a cutoff of circulating tumor DNA, again detected at baseline with a threshold of greater than or equal to 3%. And also when circulating tumor DNA was looked at as a continuous variable. And we again can see nice separation of the curves with significantly worse event-free survival in this prospective study in patients with elevated circulating tumor DNA at the time of diagnosis prior to starting therapy. This association between elevated circulating tumor DNA detected at the time of diagnosis was again prospectively demonstrated in a study from the French trial, OS 2006, where similar findings came out of this correlative biology. And this figure is a demonstration, a tool that this group developed, combining pretreatment circulating tumor DNA with clinical features that we know are important to be able to predict the risk or survival for each patient, again, combining both clinical features and pretreatment circulating tumor DNA. This was, again, prospectively collected data, and because of these two studies, we feel that circulating tumor DNA is ready to define eligibility for higher risk groups and lower risk groups in a trial if we were to be able to run and write one today. Next, I wanted to take a look more closely at MYC as a poor prognostic factor. This is a molecular feature that was mentioned a lot at this year's factor, so I know a lot of people are familiar with this, and it's discussed a lot in the literature. And so I wanted to take a few moments to review some of the salient literature and some of the challenges to translation that we're currently wrangling with. This was an older study conducted by the German group where we noted that amplification where MYC resides on chromosome 8 was associated with relapse in all cases, and that held up to be true in patients when they removed patients from the cohort that already had pulmonary metastatic disease at the time of diagnosis. More recently, this study by Danoon et al. Looking at two different publicly available cohorts, Target and ICGC from the UK, showed that MYC amplification was associated with with worse outcomes. This should be overall survival in both patients with localized and metastatic disease. This is a study that came out recently from the Shanghai General Hospital, which included an integrated multi-omics platform to look at different subgroups that differed biologically and in terms of outcome. And so they integrated copy number changes with RNA sequencing data and methylation, and they found 4 different clusters. One was immune activated, the second was immune suppressed, the third was HRD dominant, and all of these have somewhat overlapping survival, but you can see this group with significantly worse overall survival in purple, and that's the MYC-driven subgroup. This is a study that I had the opportunity to lead with Dr. Katie Janeway a few years ago, where we looked at 2 different cohorts of patients to first try and understand, our initial goal was to try and understand the genomic landscape of advanced osteosarcoma in our primary analytic cohort here to the left, which was comprised of 92 patients, about a third of which had metastatic disease at the time of the study, and then in an unbiased fashion explore which genomic features were associated with prognosis. And we found that in this cohort, MYC amplification, which we defined as a copy number greater than 7, shown in the blue curve, is associated with significantly worse overall survival. And then in a second cohort from Memorial Sloan Kettering of patients with only localized disease, we again found that MYC, here defined as a fold change of greater than 2, shown in blue again, was associated with significantly worse overall, and in this case, event-free survival as well. And then a few months ago, there was a study published that demonstrated that in a totally new cohort of about 48 patients that underwent both DNA and RNA sequencing, MYC expression was associated with significantly worse survival with high MYC expression shown in the blue curve. And so with this preponderance of data, is MYC amplification ready for prime time? And the answer is not quite. And to try and address why, how could we not yet be using this when we have so many different studies demonstrating that MYC seems to be a bad actor? I want to give a little bit more detail about how some of these studies were done and highlight some of the challenges to putting all of this together. So each of these studies, while demonstrating an association with MYC biology and poor outcome, used a different platform or a different way of testing MYC. And usually different cutoffs. So this study by Smita was using a SNP array. The Danoon study was using whole genome and whole exome sequencing. The Shanghai study was using this multi-omics classifier. Here in our study, we were using 2 different targeted NGS sequencing assays at 2 different institutions with different pipelines and platforms and different ways of actually defining MYC. And then the Van Uwijk study that was recently published looked at MYC expression by RNA sequencing, which showed a significant outcome. But they also looked at MYC amplification based on copy number using our threshold of greater than 7, and that was not associated with significantly associated with outcome. And so while it seems clear-cut, there are still several key questions that we need to address in order to move this into a clinically implementable form. So what is the optimal cut point for defining MYC amplification? What assay should we be using? At which biological level should we be looking at MYC? Is it the DNA level, the RNA level, protein, a combination of these? And we need prospective validation with a predefined threshold and statistical plan. All of these studies that I showed you, while convincing, were in retrospective cohorts. There's several ongoing studies looking at MYC in association with outcome. Sarah Whittle presented her work in conjunction with the Children's Oncology Group, looking at a case control of MYC amplification versus those that did not have MYC amplification in bank samples. And in her study that was presented in a poster at ASCO, she found that there was a trend toward worse survival in patients who had MYC amplification that was done with a SNP array and defining this as a ratio of greater than 2 between the MYC copy number and chromosome 8. And while there was an association while it looked like those patients who had MYC amplification did worse, this was not statistically significant. And Matt Dietz presented his work at Factor this year, looking at the prognostic significance and microenvironment influence of cMYC and osteosarcoma, and also using FISH as a tool to detect MYC. And as more studies are emerging, I think we're realizing this is not as straightforward as we initially thought. We, I think, have a strong hunch that there is something to MYC biology, but demonstrating that there is an association with worse outcome is not the same as translating it into a form that we can clinically use. And so I think actually more confusion is probably good, because it helps generate more questions that will help us critically think about what are really the most useful markers that we should be using, and how can we turn those into tests that can be embedded into clinical trials. I want to transition a little bit on molecular features that may guide therapy selection in osteosarcoma. This is a little bit different in flavor than the prognostic markers that we've been looking at, but we felt it was also important to take a look at molecular features that could really help us to prioritize targets for therapy. And this would be most likely initially applied in the relapsed and refractory setting in clinical trials, and ultimately, if there is signal into the upfront setting. One growing area of interest in osteosarcoma is the tumor cell surface, or what we call the cell surfaceome. These surface proteins are attractive because they're accessible and can be used not only as biomarkers, but as direct therapeutic targets. The therapies being developed include antibody-drug conjugates, or ADCs, which link a targeting antibody to a cytotoxic payload, and CAR T-cell therapies, which redirect T cells to recognize and kill tumor cells expressing a particular surface antigen. The ideal target is one that's highly and selectively expressed on osteosarcoma cells with minimal expression in normal tissue, and that specificity is really critical to avoid toxicity. Recent profiling studies have identified several promising surface targets, including HER2, GD2, LRCC15, EGFR, as well as newer players including MT1, MMP, MRC2, and CADM1. And these appear to be abundantly and selectively expressed in osteosarcoma cells. Some of these targets, like GD2 and B7H3, are already being tested in clinical trials using ADCs or CAR T cells, and there's ongoing interest in developing these. But to date, we really haven't had the home run success that has been seen with therapies like CAR T cells in hematologic malignancies. And this really could be its whole other talk, and there are experts likely in this room that can speak to this better than I. But to sort of give a sense for why do we think what do we think really accounts for this lack of slam dunk success in this arena? We think it's more than likely a combination of factors. There's probably heterogeneous antigen expression, poor T cell trafficking, and highly immunosuppressive tumor microenvironment that's contributing. And going forward, we'll need more detailed correlative analyses, really looking at both patients who respond and those who don't respond in clinical trials to really figure out which targets, therapies, and patients are most likely to benefit. And importantly, we'll need to define what level of antigen expression is truly meaningful in osteosarcoma for each of these. Another area of interest is homologous recombination deficiency, or HRD. This is one of the cell's main DNA repair pathways, and when it's dysfunctional, tumors can become vulnerable to certain therapies. Many people may be familiar with BRCA1 or BRCA2 mutant cancers, where HRD is used as a predictive biomarker for therapies including PARP inhibitors. And this has led to the FDA-approved treatments across multiple cancer types. And while BRCA mutations are relatively rare in osteosarcoma, there are HRD-like features in osteosarcoma that we think we can exploit therapeutically, including alterations in the Fanconi anemia pathway genes, RB1 loss, BRCA1 promoter methylation, transcriptional patterns, and patterns of genomic instability that resemble HRD, even without a single driver mutation. The challenge in osteosarcoma is that there's no standardized way to define or measure HRD in this disease. Where tools that have been utilized in other cancer contexts have not yet been validated. But is certainly an area of interest, both in terms of discovery and in terms of developing clinical trials, leveraging these kinds of therapies. This schematic tells us is sort of an overview of how we think about preclinical models showing sensitivity to PARP inhibitors that can then be tested in clinical trials, including one that recently completed accrual, a phase 2 trial of olaparib, a PARP inhibitor, and an ATR inhibitor, ceralasertib, in relapsed osteosarcoma. And we're currently working out the correlative studies here to, again, try and understand which patients responded and why, and which patients didn't respond and why, and can we use that information to iterate and make better trials in the next round. Again, zooming back out on our overall priorities that we've identified for translation that emerged from our cBOSS working group discussions. Again, we need to compare these classifiers. Each of them are promising individually, across the data sets where they've been demonstrated. But again, how do they link together? Are these different classifiers actually telling us the same thing? Or are they providing complementary information where we can combine a few of these to develop an even more robust and accurately predictive model? To do this, we think we need to collect multi-omics data, importantly linked to clinical outcomes to validate these stratification strategies in both retrospective and prospective cohorts when feasible. When feasible, we are recommending a shift to whole genome sequencing and whole transcriptome sequencing that are adapted to clinical pipelines. And so that means using samples that are FFPE based and developing pipelines that can accommodate that, because that's what is used in clinical pathology and will be most relevant for translation. We recognize that doing whole genome sequencing and whole transcriptome sequencing right now is not widely available and can be expensive, but as these are becoming more readily available and as the data is showing us that we actually learn more by doing some of these wider assays, we think this will be important in our correlative work and in our preclinical work. In parallel, we need to prioritize the clinical development of promising biomarkers for targeted therapies. And address key implementation questions that we highlighted in our, in particular in the MIC case study, but this applies to many of the molecular features that we looked at, including defining cut points. I think one of the elephants in the room is, of course, well, how do we actually even act on this? Even if we're able to identify patients with high-risk disease, what will we treat them with? And I'm going to try and address that in a couple of slides to try and sort of start a conversation that I hope to continue with many of you for many years to come. For now, I want to highlight some of the next steps and outputs that are already spinning out of cBOSS. We're working on our final stages of manuscript preparation for putting together the work that we've done, and hope to have that work out there for everyone to read in the next several months. And while that is undergoing edits, our cBOSS contributors are already sharing and comparing their own institutional data organically, which is really wonderful to see. We are in the process of putting in proposals for retrospective integrated analysis in bank samples, and then also working out analysis plan for prospectively collected samples on our ongoing trials. There's also major efforts toward developing standardized approaches for sample banking and molecular characterization. And we think this is really, really essential to be grounded in equity so that every patient with osteosarcoma, no matter where they're treated, can really benefit from the advances in precision oncology, and that their tumors can undergo all of the molecular characterization that would be available to anyone anywhere else in the world. This very busy slide is my attempt at trying to answer how we might think about a clinically useful model for risk and biology-informed care. And so this is certainly not the schema for the next clinical trial, but is something that we can think about and imagine together as a framework for how this might be implemented in the clinic. And I'll walk us through kind of the big picture here, and then I have a couple of cases that I hope can illustrate the principles here. And so the idea would be that all patients with newly diagnosed high-grade osteosarcoma would have their tumor samples molecularly profiled such that we would have enough information in combination with clinical features to be able to predict which patients are likely to respond to MAP and to get our standard therapy, which patients are at high risk for relapse and dying from their disease and really need different therapy that is better matched to their risk and disease biology, and which patients may have a really outstanding outcome and may not need to be subjected to the short and long-term toxicities associated with our current standard of care. And while we've discussed that we still have a ways to go in being able to clinically implement a lot of our biology features, we can imagine in a few years from now that we may have more validated molecular features, including, in addition to high pretreatment ctDNA, MYC amplification or overexpression, other copy number alterations, genome-wide loss of heterozygosity, high-risk transcriptomic signature based on RNA sequencing, or a high-risk methylation signature. And so patients with standard risk disease that have localized, non-pelvic, and resectable primaries will get our standard of care. It works well for a subset of patients. For those patients who have a favorable risk biological feature, like a favorable risk transcriptomic signature, and they don't have any high-risk clinical or biological features, perhaps these are patients that might be eligible for a de-intensified regimen. We're not there yet, but I think we can imagine that someday we might get there. And then for patients with high-risk disease, going back to that elephant in the room question, this is not a perfect answer to this because it doesn't tell us exactly what we would give these patients. But what it does suggest is that these patients need something other than our standard of care therapy, and may give us an opportunity to have these patients receive novel agents or a novel schedule on a high-risk arm of a clinical trial or on separate clinical trials. And concurrently to be able to collect their specimens at time points when they would be undergoing procedures to really learn as much as we can, so that for the next iteration we're really able to better tailor our therapy to the biology that we're continuing to understand better and better. So here's our hypothetical patient 1 case to see how this could be applied. We have a 16-year-old male with osteosarcoma of the left femur. It's localized, it's resectable. Based on what we know today, this should be a good outcome. And then we have a molecular workup which shows that pretreatment ctDNA is elevated at a level beyond 3%. DNA sequencing shows a TP53 rearrangement as well as a CDK4 amplification. RNA sequencing and methylation profiling is consistent with high-risk biology. And so this patient, which we would think, based on what we are using clinically today, would have a good outcome, has several features here that demonstrate that this patient has high-risk biology. And so perhaps in a future world, we can imagine that this patient might be eligible to receive a high-risk or novel therapy that is different than our standard of care on a clinical trial where we can continue to learn. In hypothetical patient case 2, clinically, this patient looks the same, a 16-year-old male with an osteosarcoma of the left femur that is also localized and resectable. And this patient has no detectable ctDNA at baseline, a TP53 rearrangement, and no high-risk copy number alterations. And then RNA sequencing and methylation profile is consistent with a favorable risk biology. And so perhaps this patient could be eligible for de-intensified therapy in the future, something like MAP and surgery, and then be done and not have another 19 weeks of MAP. Or maybe as we're learning more about the biology, we may learn that these patients are most sensitive to alternative therapies like immune-based therapies. Again, we're not yet there, but one can imagine how in a clinical context we might be utilizing all of these features together with our clinical features to really drive more precise patient care. In the last couple of minutes before I take questions, I wanted to highlight an ongoing effort that I'm leading at UCSF that is separate from cBOSS, but it's sort of a response to some of the gaps that we've highlighted. And some of the gaps that we've already mentioned here, but that I will summarize, include the fact that much of the data is based on small, retrospective, and heterogeneous studies that are using different platforms to study molecular features and comprised of different patient populations. It's unclear which of these biomarkers we should be using and which thresholds to use. And this is something we haven't talked about so much, but a lot of the data out that's available publicly has really great genomic and other molecular data, but is not complete with the clinical annotations. And so we have the information about what the biology looks like, but it's not linked to how the patients actually did. And so that makes it really hard to make associations between biology and clinical outcome if we're missing half of the puzzle. Many of the studies have relied on research grade platforms and fresh frozen tissue, which are really important for biological insights, but are not yet primed for being incorporated into clinical care. And of course, we've talked about lacking clear actionability. And so in a study that I am putting together here at UCSF and that I hope to expand to a multi-institutional cohort, we're going to be running samples on a single clinical pipeline, collecting multi-omics data, pairing the genomic data with really comprehensive clinical characterization, and leveraging, instead of a research grade platform, a CLIA approved or already clinically embedded RNA sequencing platform using FFPE or the sample type that the pathologists already look at when they're making a diagnosis, and to try and link these prognostic biomarkers, not just with outcome, but with novel therapies and trial designs. And so the study is called Liftoff. Every good study needs a good acronym, leveraging insights from transcriptomics for osteosarcoma using FFPE, with the goal of validating transcriptomic prognostic biomarkers in newly diagnosed osteosarcoma, really with the goal of translating these into clinical stratifications. This is a little bit of a misnomer because it's not just focused on the transcriptomics or RNA sequencing. We'll be combining this with DNA sequencing as well. So we have a pilot cohort at UCSF using FFPE and a clinical assay called the UCSF 500 that is a targeted DNA sequencing assay, and then a whole transcriptome sequencing assay with return of results currently for fusions, but we get expression as well. And so we're using this platform that's already adapted for and embedded in clinical care to try and validate these prognostic markers that have been demonstrated in sort of siloed and primarily research grade settings. And so this is an opportunity that I hope will be not just a focus at UCSF, but can expand more widely, and that other initiatives like this will help us get from where we are today to really being able to implement these in clinical trials and clinical care. So in summary, cBOSS offers a structured framework to assess the translational readiness of our candidate biomarkers. And there are a lot of promising classifiers, but even those like MIC require refinement, validation, and standardization. And our progress really hinges on shared infrastructure, prospective data, coordinated efforts, all really embedded in a framework of collaboration and equity. And there are multiple initiatives that are now moving these classifiers toward implementation. And with that, I'd like to thank the entire cBOSS group, especially Ryan Roberts and Andy Livingston, who really spearheaded this and trusted me to help lead the group, to MIB agents, really, for all you do to make it better, and Christina for inviting me today, and to the Osteo Angels and Osteo Warriors and their families that inspire this work and make it possible to make it better. And with that, I'll take any questions.
Thank you so much, Dr. Marinoff. And yes, if you have any questions, please add them to the Q&A, and we will share those with Dr. Marinoff. But I just want to say, first of all, a couple things. It makes me so happy that this was sparked by a conversation at Factor, which is kind of what Factor is all about. And I so admire this effort because this is such a difficult effort, these large collaborative group efforts, especially this is truly a global team. And so, thank you, and to all the consortium participants, because it's in your spare time that you all have with all your other responsibilities, it's really difficult to move projects like this forward. So thank you for all your efforts on this. So I want to kick off with a couple questions. So, and you kind of touched on this in this very comprehensive presentation. Thank you. But it seems like, getting to the next steps with these biomarkers, a couple things, like you obviously need more data. And so it sounds like you're definitely trying to figure out how to bake in these standards for collecting this type of data in future studies and future clinical trials. So I guess, first of all, a question around that, just kind of how you do intend to kind of implement some of those standards so that moving forward, you kind of are able to collect as much data as possible in studies and all these different, trial networks, for example. But that also, in order for this to be implemented clinically, those tests also just have to be available to patients at different hospitals and institutions. So some of these tests right now that they might be kind of maybe more limited into kind of where patients can get those done, but ultimately they would need to be, I guess, available and cost effective and covered by insurance even for it to be implemented clinically. So want to get your thoughts on that. And also specifically a couple questions that came in, for example, liquid biopsy for ctDNA. I think that was noted as like ready for clinical implementation, but is that a standard test of diagnosis? And if not, how do we get there?
Really excellent and important questions. I'm going to start with Allie's question around ctDNA, and then I'll go to your bigger picture questions, Christina. Liquid biopsy for ctDNA based on the prospective leopard study that I showed you and the French study, we think is ready for prospective evaluation. And that is different from it's ready for a patient who walks in today into the clinic where we can send the test and know how to interpret it and how to act on it. I think we will get there, but the next step in this process that I think we all wish were faster, but also it's really important to do as rigorously as possible because it's going to affect our clinical decisions for patients. And so I suspect that for the next iteration of clinical trials with upfront osteosarcoma, we will incorporate circulating tumor DNA potentially as a high risk risk defining feature, but we're not yet there. And while there are some commercial assays available for liquid biopsy for ctDNA for patients, it's not something that we're routinely offering because it's still in a research space. And this question and this hope that we can take this information and act on it in a way that will clinically benefit a patient is something that, again, a place I think we'll get to. But if we got a circulating tumor DNA level right now, I think the standard of care, which is absolutely appropriate, would be to do our standard of care. And so we're really not at a place where we can say we've got an elevated ctDNA, let's do something other than MAP, because that hasn't been tested to be safe or effective yet.
I know I asked you, I think probably like 10 questions in one in that first question.
Yeah, let me try and tackle them.
Yes. So the other question that came in was, I think you're pulling up that slide about the blue squares.
Yeah, we can do the blue squares and then go to your bigger question about how do we make sure all patients benefit from this, and how do we what really needs to come next to turn the blue squares and the yellow squares and the red squares into green squares, I think was sort of the gist of the question, if I'm getting that right. Allie, your question around our blue squares intended for clinical trials. I think they're in a space where they've piqued enough interest, they've been reproduced in enough retrospective cohorts that they will be tested in some capacity in both prospective and retrospective cohorts, including using samples that are collected from patients on clinical trials. But that doesn't mean that they're yet ready to say a patient is going on a clinical trial at the time of diagnosis. We know that they have these alterations, and therefore we can stratify treatment based on them. We're really at the point where they could be where samples could be collected for a patient that's on a trial and then studied later on to understand how things like MYC amplification and CDK4 amplification impacted survival in a clinical trial setting where we know how the patients were treated. We have those complete clinical annotations, and there was sort of the control over the trial that one benefits from in a trial. And so I think we will be testing these as we are able to access more samples from patients who are on trials, leveraging bank samples from institutions and from cooperative groups, but we're not yet at the point where we would yet put a patient on a clinical trial and say that they have MYC amplification, and therefore we can direct our treatment based on that yet.
And Dr. Marinoff, so going back also to the standards, so will you, or you may have already done this, or will you be publishing what those standards are? So you had mentioned like the WGS, WTS, the different types of multiomics, so that when someone is starting or designing a clinical trial, whether that's a COG trial or another clinical trial network, or even just, a PI initiated trial, that they can kind of look at, what are all the pieces of data that we should be collecting and making sure we're budgeting for that correlative study as part of the trial so that we can collect this information.
Yes, cBOSS is really meant to be that kind of resource where we're able to put a spotlight on a subset of molecular features that we think have promise and need further testing, and not just sort of the broad, like, we need more testing, but really the why and the how, and which assay and what cut point and how should we be doing this. And importantly, it really needs to be done in conjunction with our statisticians, because this challenge of identifying which cut point should we use to define criteria is one that's really tricky, and requires the expertise of a statistician based on the cohort that we're using and the design to help us really be able to pinpoint those thresholds that are required to be able to make a test clinically useful.
Yeah, that's great. That's fantastic. So it'll be like true, just implement, like, we can do this.
That's really the goal, and I think it's not so far-fetched. It's a first step, but we hope it'll be a resource for everyone, and really just a version 1. The hope and expectation is that there's going to be more and more emerging molecular classifiers and tools that are available, and that will add to this work, which is the sort of the state of affairs of a snapshot today. To get back to your question, Christina, of how do we make sure that everyone has access to these kinds of assays and testing, and that we can bring the promise of precision medicine to everyone, and fill in those gaps of translation, I think a lot of things need to happen, and that's sort of why this slide is so busy. And a few words here encapsulate a lot of pretty extensive concepts. So retrospective and prospective cohorts, that's like a lot of words that mean, in my mind, that we really, with osteosarcoma as a rare disease, we need to be leveraging as many samples as we can, and collaborations as we can. It's wonderful to see a number of collaborative efforts emerging, not just cBOSS, but was excited to learn about Foster and a few initiatives coming out of the UK, and other efforts really bringing together multiple institutions and sometimes multiple countries so we can pool our data, our samples, and then turn it into a form that we can utilize and say, okay, this cohort used the apple assay, and this one used the orange assay, and marked things using the pear language, and this one used the grape language. How do we actually harmonize that so we can make sense of this mixed fruit bowl to actually utilize it? Because the real world data is never going to be perfect, but that really shouldn't stop us from squeezing the most out of it as we can. And so I think we really need to put in the efforts to study all of the samples that are going to be incoming, but also are that available, and again, making it usable. So not just the formatting the data and structuring our statistical plan that makes sense for the data that we have, but again, linking it to clinical outcomes so that we have the other key piece of the study. One way that we think we can do this for patients who are sort of in real time being diagnosed with osteosarcoma is an effort that is held at the National Cancer Institute called the Molecular Characterization Initiative, that has been initiated for some rare cancers. Ewing sarcoma was recently added to this, along with soft tissue sarcomas, where every patient, no matter what hospital, can have their samples sent to the NCI and have them undergo multi-omics testing. And that, I think, is a really critical way to ensure that all patients are getting equitable care, because it's unreasonable to expect that every single facility is going to be able to have an assay like the one I mentioned, like the UCSF 500 with whole transcriptome sequencing. And yet every patient, if we decide that these are tests that are clinically useful, should be able to benefit from that. And so that is one effort that I think we could really benefit from, and there's lots of us working to advocate for that. And I think folks from this group also, coming from all different perspectives, probably could be really instrumental in that effort.
Yeah, that sounds amazing. And one more question before we let you go. So you guys did this very extensive review in 2024 of kind of all the what's been published and possible biomarker candidates. And so what is the framework for moving forward as people continue to make new discoveries and their new biomarker candidates? Is there, just kind of an annual review? Is there a way that people can kind of submit candidates?
That is an amazing question, and we haven't yet figured out the exact what's next, but I really love that suggestion. This needs to be the V1 that can continue either as the current cBOSS or sort of the next a next different form of this. So I really appreciate that suggestion, and we'll put my head together with the rest of the group to figure out how are we going to make this living and breathing and changing with the data that's emerging.
Well, you've made so much progress to date over such a short time period, because again, to get kind of everyone working together, go through that tremendous review and get consensus, and also kind of just take the steps that you are towards, again, just kind of group implementation is fantastic. So again, thank you so much for your efforts. And thank you so much for joining us on OsteoBites today and for making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on our website at midagents.org and on our MID Agents YouTube channel and at your favorite podcast place. And then we hope you can join us on OsteoBites on August 28th. So in just a few weeks, we're going to be joined by Dr. Brian Ladle from Johns Hopkins University, and he's going to be discussing his lab's work on cGAS pathway studies in canines with osteosarcoma. You can find our OsteoBites lineup for the next few months on our website. And if you ever have any ideas for topics that you'd like to hear about, please email us at events@midagents.org. Thank you again so much, Dr. Marinoff, for spending time with us today. And thanks to all of you for taking time out of your busy day to join us. We hope to see you back here in a few weeks when we chat with Dr. Brian Ladle. Thanks so much. Thank you, Dr. Marinoff.
Thank you, Christina.